due to COVID-19 disease outbreak and all, we are not in a situation to go for classroom teaching, and hence we are trying to adopt situations wherein we can have online deliverance of the classes, and we are adopting one of such kind. This is a class on plant physiology meant for MSc Botany students, MSc Plant Science students, and the text what we follow in this case is the text authored by Theis and Zeiger, Plant Physiology. And here we will be first going with the chapter on plant growth hormones, and here we will start with the topic on auxins. And how we proceed is that we first have a brief description on the history of how these hormones were discovered, and then we move into the biosynthesis aspects of these hormones, and further we will go into the physiological role played by these hormones, and at last we come into how these hormones bring about the action, hormonal action is imparted. So coming to the case of auxins, as you see from here, there are a set of experiments which have led to the discovery of auxins, and one of the first is where Darwin has experimented in the year of 1880. Here they have experimented on intact seedlings of oats, and there you can see once the light is falling from a particular direction, the coleoptile is bending towards the light. But as and when the tip of the coleoptile is excised off, you can see there is no effect on the coleoptile bending. And again, in a situation wherein the coleoptile tip was covered by a cap, again you can see that the coleoptile is not bending even if the light was falling onto it. In another set of experiments by Auxins and Jensen, they experimented in such a way that towards one side the light was falling, and towards the opposite of that they inserted a mica sheet, and there was no effect on the bending of the coleoptile tip. But when the mica sheet was inserted to the side where the light was falling, this was the same side where the light was falling, and you can see that the coleoptile was bending towards the light. So some sort of informations were deriving out of these sort of experiments, although you feel that it may be very simple experiments, but that have brought about significant contributions to the discovery of auxins. Again, the repetition of some experiments were conducted by Auxins and Jensen in the year 1913. Here you can see the tip was excised off, and in that place a particular gelatin was inserted, and even if the gelatin was inserted, still there would be percolation of the substance what was there in the tip to the down portion through the gel, and once it reaches down you can see the same effect of light was performing. So that means water substance which was bringing about the bending effect was percolating through the gelatin and reaching down. And coming to the next aspect, again it is set of experiments conducted by Paul. Here they have excised the tip of the coleoptile and just replaced it towards the other side, to the other end, and they tried to give light from one point. Again the light was falling from this direction, and you could see that even if the tip, one portion of the coleoptile tip was placed, that was enough for the percolation of the material which was in the tip region to go down and result in the bending of the coleoptile. Again, the experience was further taken ahead with the experiments of Wendt, wherein they could excise the tips and they put it on the gelatin, and they allowed the material what was there in the tip to percolate down, and once it had percolated down, they just excised the gelatin from there and kept it on the tip of the coleoptile, and this placement of this gelatin tip was enough to bring about the curvature once the light was falling. So that means the tip was not necessary for the bending. Whatever material at the tip of the coleoptile which had percolated down into the gelatin tip could bring about the bending of the coleoptile. So these were some of the set of experiments which was conducted in the last century, and wherein they could clearly bring about the effect that there was something at the tip of the coleoptile which was percolating down, and that was bringing about the bending effect once the light was falling. But at that time they were not clear that auxin was responsible for that. They just identified that there is some chemical substance which were oozing or percolating from the tip of the coleoptile, and which had the effect of this particular effect. Now we go into the next aspect of we will try to learn something from the discovery of auxins, or they came to a conclusion that there was some chemical substance, and further analysis of this chemical substance gave the information that it was a chemical structure which brought about, or chemical substance which brought about this effect, which had a structure with these peculiarities. That is, it will be having a planar aromatic ring acting as the binding platform. Secondly, the chemical structure of this substance would be having a carboxylic acid which is acting as the binding site. And third, it would have a hydrophobic transition region separating the two binding sites. So here we have the planar aromatic ring acting as the binding platform, a carboxylic acid towards the end of it, and there was a hydrophobic transition region separating the two binding sites. So any chemical structure which could fulfill these three aspects or these three peculiarities can perform like an auxin substance. That was the conclusion from this discovery. And together with that, they also came out with the invention of anti-auxins. Anti-auxins are substances which have no auxin activity, but they can compete with an auxin. For example, IAA, that is an auxin, indole acetic acid, for specific receptors without triggering auxin response. So they will just go and bind, but they will not bring about any auxin action. So they can, in one way, can prevent the auxin activity. That is the specialty of these antioxins, and one such antioxin is alpha para-chlorophenoxy isobutyric acid. Coming to the biosynthesis aspect of auxins, it would be better we go directly into the pathway. I always suggest that the book which we are following for plant physiology, that is Trace and Sager, it is better either to have an e-copy or a hard copy and follow it closely with this, because it may not be clear from the slides what has been shown here. So if you have any copy or a hard copy, it will be better you keep it open and try to follow with that. Whatever we try to depict here, whatever we try to project here is everything which has been taken from the textbook of Trace and Sager on plant physiology. Here you can see the indole, this is the indole 3-pyruvic acid pathway, and here it starts from tryptophan. This is the precursor of indole 3-pyruvic acid, and tryptophan can proceed in 1, 2, 3 and 4 different pathways for the synthesis of IAA. And how it proceeds? This is the first pathway. Okay, this is the first prominent pathway, that is the IPA pathway. Here there is an intermediate called indole 3-pyruvic acids. So tryptophan gets converted into indole 3-pyruvic acid, then it gets converted into indole 3-acetaldehyde, and finally it gets converted into indole 3-acetic acid, that is IAA, one of the auxin. Similarly, there is another pathway which is called the TAM pathway, that is directly tryptophan gets converted into tryptamine, then tryptamine gets converted into indole 3-acetaldehyde, and finally to IAA. And this is ion pathway. So this is IPA pathway, this is the name which has derived. This is the TAM pathway from an intermediate, again based on another intermediate IAN, that is indole 3-acetaldioxime. This is another pathway wherein again it culminates in the synthesis of IAA, and again we have another pathway, that is a bacterial pathway which is followed in the case of bacteria. This is again another pathway called as IAM pathway. We have an intermediate called indole 3-acetamide. So, these are the 4 different pathways by which the indole acetic acid is synthesized from the precursor called tryptophan, and the description regarding this is given over here. In dividing tissues, shoot apical meristems, young leaves and developing fruits, these are the main areas where auxins are being synthesized. The first we have already described, IPA pathway is a deamination followed by decarboxylation and then oxidation by specific dehydrogenase. That is what happens in that particular pathway. Second is TAM pathway. Here we have a deamination and a decarboxylation is reversed, and different enzymes are involved in this pathway. Third is ion pathway, mainly identified in Cruciferaceae. Then we have in Graminae, Musaceae. This is the prominent pathway. And the last ion pathway, the intermediate is indole 3 acetamide. It is used by various pathogenic bacteria such as Pseudomonas sevestoni and Agrobacterium. Now, I am going to describe to you regarding one of the important features with regarding to auxin, and this is the only growth hormone which is being transported polar. There is only polar means of transport for this particular growth hormone. Only plant growth hormone that is transported polarly. Polar transport contributes towards auxin gradient from shoot to root, which affect various developmental process. So, it is being synthesized in the shoot and moves down into the root, and including stem elongation, apical dominance, wound healing, leaf senescence. These are very important physiological functions which is being taking place in the plants, and this as auxin has got role in this. Polar transport proceeds from cell to cell rather than symplastic. So, it moves from one cell to the other cell, and high energy requirement is there in this particular process. Let us see how this polar transport takes place. This was first proved by a simple experiment wherein they tried to excise out a piece of hypocotyle, and the tip end or the apical end was named as A, and the basal end was named as B. And 2 donor blocks were kept, one a donor block and another one is a receiver block, and containing radio labeled auxin. This is for identifying that they are using radio labeled auxin. And in this case you can see whatever radio labeled auxin was moving down to the receiver block without any hindrance. And they could easily identify this auxin because it is radio labeled. Whatever got collected in the receiver block, they could further identify and see that the auxin has moved from the A tip towards the B. But when this was reversed, here this is a case where it was inverted, and here you can see the donor tip was at the top and the receiver was at the down, and here you can see the transport of the auxin was blocked because it can only move polarly from one end to the other end, not in the reverse way. And I will be explaining more on that why it is happening so from this particular diagram. Here we call it as chemiosmotic model was proposed for the polar transport of auxin. Here you can see only towards one end of the cell in the membrane there are transporters for IAA, not everywhere. It is not towards this end, but only towards this end. You can see the lower end only, these red ball structures are basically the transporters of auxins. So, once the auxin entering in this particular pathway, the auxin can enter in 2 different ways. One is the IAA can link up with proton and form IAH and then can move inside. And the other way of transport is a symport system wherein it moves along with the protons. There it do not need to combine with IAH, but it can enter as an anion, but it can go along with a cation only, that is a proton, and both simultaneously move in. We call it as a symport system, and for this energy requirement is there. As you see from here, the ATP is utilized for the protrusion of the protons, and once the proton gets accumulated in this area, in the cell wall area only, it can come through these transport proteins inside, and during that particular transport IA is also getting transported. So, by these two means IAA is getting transported into the cell. But once the entry of the IAA is possible, the exit of the IAA can be only possible through these transport proteins, what you see as red over here. These transport proteins will take IAA minus, that is the anion, and it will transport outside. So, when you keep this upside down, there are no transport proteins over here. That is why it is not able to move upside down. When the tip is kept upside in this particular case, you can see when it is inverted, the transport proteins will be always remaining at the top portion, and the lower portion there will not be any transport proteins as you see over here. So, only IAA can move down in this particular path because of this transport proteins, and we call it as chemi-osmotic model, because here there is protrusion of protons, built up of proton gradient that will help in the movement of IAA through a transport protein, and through which IAA is entering inside the cell and IAA exit out through the particular transport proteins polarly. This is one of the specialty of auxins, because you cannot find this particular situation in any of the other hormones what we are going to describe ahead, cytokinins, gibberellins, ethylene. This is not the means of transport. This is a unique way of transport what we see in the case of auxins alone. The rate of polar transport is very slow, but still it is 10 times faster than diffusion. If you see from here, it is approximately 1 centimeter per hour. Why it is very slow? Because it has to enter inside the cell through a import system, and it has to get exited out through a particular transport proteins polarly. So, this movement takes place through the help of two different proteins, one for the influx and one for the efflux out from the cell. So, this is comparatively going in a very slow process. That is why it is being shown as 1 centimeter per hour. So, the polar transport of auxins require protein carriers that can recognize the hormone. We have already seen that at the lower portion we have specific protein carriers. So, that can recognize this auxin and can help in the efflux of this protein from the cell. And inactive auxin analogues and auxin metabolites are not transported polarly, because this can only recognize the auxin and it will help only in the movement of auxin out, but not in the case of any auxin analogues or not in the case of any auxin metabolites. Now, coming to the roots. So, this is generally what happens in the shoot. So, you will be always wondering what is happening in the case of roots. In the case of roots, there is acropetal transport. Once it reaches to the root region, it can move towards the tip of the roots, that is acropetal transport. And small amount of basipetal is also happening, transport is also happening, that is from the towards the base region, that is the basipetal auxin transport is also noted. But major transport, once it has reached to the root portion, the movement of the auxin will be acropetally from the end of the shoot towards end of the stem towards the tip of the roots. Now, the auxin transport can be inhibited by certain inhibitors, which can specifically block the auxin efflux. And where does it block? You have already seen that we have the exit proteins, what you see in this red. So, if something which can bind over here competitively to IAA, it can bind, but it may not get transported out, but it can still prevent the further binding of IAA towards that, such a competitive binding. Because of this binding, what happens is that further IAA will not be in a position to bind, and two such chemicals are being explained here. One is NPA, another is TIBA. And both of these chemicals, these are supposed to be inhibiting the auxin action, because they can bind to the efflux protein, and at the same time they will not allow the auxin to bind to that. So, that auxin action can be prevented, because auxin will not reach to the regions where it is supposed to. And only when auxin reaches the particular portions where it is supposed to, then only we can see the auxin action. So, that is being prevented. It causes stunting, inhibition of root growth, loss of gravitropic and phototropic responses. So, these are the basic physiological functions which are being fulfilled with the help of auxins. And once these inhibitors of NPA and TIBA binds to that, you can see the stunting of the plant take place. Root growth is getting inhibited, then gravitropic and phototropic responses also will not take place. The TIBA inhibits polar transport directly by binding to the auxin binding site on the efflux carrier, that is the red carriers what you see at the lower end of the cell. In contrast, phototropins bind to an associated protein that regulates the efflux carrier. So, this is another inhibitor. In contrast, phototropins, they can bind to an associated protein that regulates the efflux carrier. So, this is another inhibitor called phototropins. They will not directly prevent the efflux carrier, but they can prevent the protein that is regulating the efflux carrier. So, either both case, basically it is going to inhibit the auxin action. Again we will be having a doubt whether the auxin is moving only polarly, because there are long distance transport has to take place in the case of big plants which are huge in length and all. So, how it is taking place? That is being explained here. Auxin is transported non-polarly via phloem. This is comparatively taking place at a greater speed and is passive, no energy is required, and the plant may be preferring for long distance transport for controlling process such as cambium division and branch root formation. So, when the plant requires auxin for these purposes such as cambium division, branch root formation, the plant may be preferring the phloem for long distance transport. So, it is not only polar transport, it is also happening through the phloem, and it depends upon the function which the auxin has to deliver. If the function is to deliver the purpose of cambium division, if the function is branch root formation, then the movement is through the phloem, not through the polar transport. Polar and phloem transport are not independent. Again, you have to understand that these are not independently taking place. They are a dependent process. For a particular distance of time, it may move through the phloem, and once it reaches the site of action, further the movement would be possible through the polar transport. So, it is a dependent process, not totally independent process. Here I am going to explain about one of the major effects of physiological effects of auxin, that is with regarding to the cell elongation. When you apply auxin to a plant tissue or to a plant, you can see the elongation taking place, stem elongation, root elongation and all taking place. How it is happening is being explained here. Here you see that once auxin is being applied, you see here IA is being applied, what you see in this blue, it comes and binds to a auxin binding protein. This is the receptor protein, and once the binding takes place, it can act in two different pathways. That is, one, it is explained as activation hypothesis and synthesis hypothesis. Activation hypothesis is that already existing ATPase on the membrane are getting activated. Because of that, proton extrusion takes place to the cell wall region, and synthesis hypothesis explains that the ABP on binding with IAA, they can trigger the synthesis of new ATPase, and that is what seeing here. Secondary messengers are formed, it moves into the nucleus, and you can see that H plus ATPase gene is getting activated, mRNA is synthesized. Coming to the endoplasmic reticulum, new ATPase is getting synthesized through the Golgi bodies. The ATPase is synthesized and delivered to the membrane. So, either the existing ATPase on the membrane is getting activated as per the activation hypothesis, or the new ATPase are being synthesized and being delivered to the membrane, so that that can become functional. In both of these cases, the protons are being extruded because ATPase is getting activated. As a result of that, proton is getting extruded. Once protons are extruded into the cell wall regions, you can see there is an acidic environment is developed over here. That can culminate in the activation of certain proteins, and one of the prominent group of proteins which get activated are expansin proteins. And expansin, from the name itself, it is very clear that it helps in expansion. One of the basic protein which can help in the cell expansion is expansin. And the synthesis process, activation process of expansin is getting activated. As a result of that, cell expansion, cell elongation takes place. So this all started from the application of auxin to the tissue or to the cell, which can result in binding to the auxin binding protein, which can help in the activation of the existing ATPase or can culminate in the synthesis of new ATPase. Whether the existing ATPase or new ATPase, it will result in the production of proton, extrusion of protons towards the cell wall, and more of protons in the cell wall region. You see here there are a lot of protons being extruded, H plus is extruded, and once we have more of H plus, the activation of expansin takes place, and more of expansin means the cell expansion takes place. So this is how one of the basic effect of auxin, that is the cell elongation, which is happening. So here you can see here auxin promotes growth in stems and coleoptiles, but it can inhibit growth in roots. So this is the way how it is promoting the growth in stems and coleoptiles, but not necessarily in roots. In roots, what we have seen is that auxin can inhibit the growth. And you will be always having a concern, what would be the optimal concentration of IAA for bringing about this growth effects. And most of the concentrations what has been worked out is that a concentration which is between 10 to the power of -6 to 10 to the power of -5 molar is optimal to bring about action of auxins. Anything beyond that or anything lesser than that can result in the inhibition of auxin effect on the plant cells. No growth will be taking place. And now we will be continuing the rest of the class in another session. For this time being, we are stopping in this session.